So today we are going to see another type genus under Hepaticopsida. This is a leafy liverwort. We had seen thalloid liverwort, Ribolia, in the last class. Now today we'll be seeing a leafy liverwort, porella. This is another type genus which is very common in tropical and semi-tropical areas. Uh, this belongs to the order Jungamaniales and Porelaceae, a single family Porelaceae. You have quite a few species commonly found in India, Platyphilla, okay, and Navicularis. So it is cosmopolitan. That means it is distributed throughout wherever there is a damp condition and conditions favorable for the growth of bryophytes in general. So as the leaf, as the name suggests, the body, the vegetative body of the bryophyte is leafy. You have a main axis, which we can call it stem. It is the main axis, which bears the leaf-like structures. There are three different kinds of leaves. The dorsal surface has a different structure. Ventral surface has a different structure. So we can call it a dorsiventral vegetative plant body. Dorsal side is more smoother. Ventral side has rhizoids and different uh, kinds of leaves. That is, leaves in different shapes, and they form rough texture on the ventral side. So we can see means you can see antheridial branch and archegonial branch. Archegonium, you can see this is an archegonial branch actually, which is enclosed in a periantha. Okay, which is enclosed in a cup-shaped perianth. So you can see this is a female plant. The porella is dioecious. You have male plants and female plants. What you are seeing here is the female plant with the perianth enclosing the developing archegonium. You can see the dorsal surface of the plant body. Okay, and then the ventral surface, you can see three different kinds lobules. Okay, you have the uh, bigger leaf, dorsal leaf, and the ventral leaf, and the smaller one, which is called the lobule. You can see it in a much better detail in the next few slides. So this is what you're going to see today. So this is how it looks in natural habitat. You must have seen whenever we go in for hill stations near the rocky areas, near the waterfalls, you can see this bryophyte growing. Porella is the only genus of this family. Grows in both temperate as well as tropical regions, mainly, as I already told you, in the mountains. So they are quite big in, they grow quite longer, 15 centimeters, and greenish texture. Okay, so the leaves of the plant are divided into two oval lobes. Upper lobe is bigger, lower lobe is smaller. And then on the third kind of leaf is also seen, which is called the lobule. So leaves are the main absorptive organs. Okay, and you have rhizoids on the ventral surfaces, and you have some amount of differentiation in the stem structure. In the older stem structure, you have some amount of differentiation, where the main function of the rhizoids is to anchor the plant with the substratum, as with other bryophytes. So occurrence, you can see it growing in moist rocks, on tree trunks. Okay, wherever you find moisture, sustainable moisture for a long time, there should be a moisture content. There you can find this bryophyte growing. And the plant body is a gametophyte, so you can divide it into stem, leaf, and rhizoids. So stem, you have a dorsiventral stem. Okay, and you see branching also, but strictly it is a monopodial branching. It does not show dichotomous branching as you saw in thalloid liverworts. What you see in Markantia, Lunularia, and Rivolia, you don't see dichotomous branching. It is chiefly monopodial branching. and they are bi or tripinnately branched. So you can see pinnate kind of structure. And the basal and the older part of the plant dies, but the growth continues at the apical region. So just like what you saw in Ribolia, you have a particular apical region through which the growth continues. Okay, the dorsal side is exposed to light, but the ventral side is exposed to the in contact with the substratum through the rhizoids. So leaves, they have large dorsal leaf. This is dorsal surface. You can see the this is the ventral portion. Okay, so these are the large leaves, dorsal leaves. They are otherwise called lateral leaves. They are oval in shape. Okay, and the dorsal leaves are bilobed. Okay, and the two lobes are, they seem to be unequal, one smaller than the other. And the lower lobe is, this one is the lobule. This is the lobule. It is called the lobule. The lower lobe is smaller, whereas the upper lobe is bigger. Okay, and that's why you say these two are unequal. The lower lobe is smaller and is called the lobule. Upper lobe is larger. So lobule, uh, suppose you have some species which grow as epiphyte over other plant genera. So they say that this lobule is modified to contain to store water, moisture. Okay, and you have the ventral leaf which is attached to the main axis. Okay, so you have three kinds of leaves. And the arrangement of the leaf is termed as incubus. Incubus, Incubous. That means the top portion of the leaf is found on the under surface of the previous leaf. So each leaf is overlapping each other. Each leaf margins, they overlap each other. So they are termed as incubus. Okay, and you have rhizoids which are chiefly smooth walled, arising from the base of the ventral leaf. This is the ventral leaf. So on the ventral side, you see a differentiated structural aspect of porella. When you take a internal transverse section of the thallus, you can see not much of differentiation is seen. The stem, actually the leaves are just one cell in thickness. Okay, they are very, very simple. They have the leaves do not have midrib, and each cell is polygonal and it is filled with chloroplast as well as oil bodies. Okay, and that is about the leaf, very simple structure. And you have the intercellular thickness is seen. This is called trigone. Okay, the intercellular spaces are thickened, which is called trigone. The stem is very simple. The young stem will not be lined by epidermal cells, but the older stems are lined by 2 or 3 walls of epidermal cell. So the you call it outer cortex and the central medulla. The outer cortical cells are small and thick walled. The inner medullary cells are large and thin walled. And the superficial stem cells of the stem give rise to paraphylls, which are filamentous outgrowths. You can see them here, paraphylls, which are filamentous outgrowths. Okay, these are the paraphylls, and they supplement the photosynthetic activities of the leaves. Okay, so young stem will not show a distinction between outer and middle, whereas the older stem shows a well differentiated distinction between the outer thick walled cortex and middle layer of medulla, which is made of thin walled cells. Okay, so this is the internal structure of the thallus of the stem and the leaf. When you come to the life cycle, we see the reproduction. You have vegetative reproduction and chiefly the sexual mode of reproduction. Then the plant is a gametophyte. Vegetative reproduction, as in other bryophytic genera, fragmentation plays a major role. This particularly porella has a high level of regeneration. So even if they are dry, even if you can preserve porella as a, you can dry it and keep it for a longer time. Whenever they, you put it in water for some time, they have the power to regenerate into a new body, vegetative body. So the basal older part of the thallus die and decay, and the branches get separated, and each branch grows to give rise to the new plant. And some species, they show discoid gemme, but this has not received very good evidences. Some of them have worked with porella, or they have seen gemme like bodies which are formed on the leaf margin, very very small, containing of one or two cells. But this is not very much in, you know it doesn't have much of evidence for this. But some of, one or two workers have done some, scientists have done some work on this, and they have seen gemme like bodies. And tubers are also, they also give rise to a new plant, which are modified special underground branches. They are formed at the end of the growing season, and when the favorable conditions arise, a new thalloid body can arise from the tubers, which are chiefly underground, under the substratum. Sexual reproduction is very well seen in this genus. As I told you, the plant is dioecious. You have male plant and female plant. The male plants are smaller in size than the female plants, and the antheridia develop actually as a particular branch. In place of a branch, you can see the antheridia developing. So antheridia are much smaller. Okay, they are adjacent to the leaves. The leaves adjacent to the sex organs are called the bracts. So in the axil of the bracts, you can see the antheridia developing. And uh, these are actually, they take the place of the lateral branches. At the time of sexual reproduction, the antheridia replace the lateral branches. So this is called the antheridial branch. So when you see the antheridium, it has a stalk, it has a long stalk. Okay, so this is the antheridium, you can see it. It has a stalk at the axil of the bract, and it consists of a globular body, a spherical body. You can see it here. When TS of the antheridial branch, this picture shows the TS of the antheridial branch. You can see the main axis, and you can see the globular antheridium. Okay, and this is the capsule, the covering, and the androcytes inside. So when you talk about, when you want to know the development of the antheridial initial lies close to the apical cell of the male branch. So the apical cell, okay, where of the male branch, which gives rise to the antheridial branch. So that divides into two. You can see the transverse division to form a basal cell and the antheridial cell, basal cell and the upper cell. The upper cell is going to form the antheridium, androcytes you call it. Okay, so antheridial mother cell you can call it, and the basal cell forms the stalk. So it undergoes, the both the cells undergo both vertical and horizontal divisions. Okay, both in the second picture you can see both the cells, upper cell and the lower cell, basal cell, they undergo horizontal and vertical divisions. And slowly the stalk cells or the basal cells surround the upper cell. You can see in the third picture, the stalk cells undergo periclinal and anticlinal divisions to form a cover, coat, jacket-like covering in enclosing the uh, antheridial mother cell, or the primary androgonial cells, they are called. Okay, so you can see it slowly encloses the antheridial mother cell, and it is called the primary androgonial cell. So the outer cells, they act as jacket initials. Okay, and in the next picture you can see them dividing further, okay, to form one layer, single layer jacket cell. And the antheridial initials, they undergo further divisions to form what is called the antheridial, androcytes. They are called the androcytes. So The jacket cell forms the antheridial wall, okay, and the inner cells, the middle portion, they form the anthrozoids or the sperms. Okay, so the jacket is more celled in thickness in the basal portion and single celled in thickness in the upper portion. So you have the stalk, it's quite long, and once the conditions are favorable, the each androcyte mother cell produces two androcytes. So the middle portion is filled with androcytes, and you can see in no time they produce sperms, which are otherwise called androcytes. So each sperm is biflagellated. It has two flagella, and the jacket bursts due to swelling of the mass of androcytes. Anthrozoid or the sperm, they are liberated in water, and the sperm swims to the archegonium. So this is how the male plants look like. The antheridium develops and produces the sperm. So when the antheridial wall bursts and releases the sperm, it looks like this. It releases the sperms in water, and they are ready to swim to the archegonia. Now female plants are larger in size when compared to the male plants. Okay, and archegonial arc is present at the apex of the archegonial branch. Okay, and the archegonia bracts form, and in this is the bract, actually involucre of bract it's called. That is a union of the bracts in such a way that they form a boat shaped structure, an involucre, which a cup shaped or a boat shaped structure at the top of the female shoot. It's called rosette involucre. It is otherwise called the campanulate perianth. It is otherwise called campanulate because it is in the shape of the cup or boat. So it is called campanulate perianth. So it encloses the developing archegonium. You can see the archegonia, and it is further after the fertilization is over also, it encloses the sporophyte, developing sporophyte also. So the capsule will emerge from the perianth, the matured sporogonium. Actually, after the fertilization, when the sporogonium is produced and the sporophyte is produced, the mature sporogonium emerges from the perianth. Okay, and archegonium, as you have seen in other bryophytes, it consists of the neck and the venter. And development of the archegonium is similar to what you saw in the previous section for antheridium. The archegonium develops again from the apical cell, and it divides into the basal cell and the archegonial mother cell. So basal cell forms the stalk. The archegonial mother cell again divides into three. Okay, divides vertically into three. So primary axial cell and the peripheral cells. Okay, the peripheral cells are going to form the outer cover layer, or the no, they are going to form the neck portion. Okay, the peripheral cells are going to form the neck region of the archegonium. Okay, and the basal cell, and they undergo vertical divisions, periclinal divisions and anticlinal divisions in such a way that they enclose the, they elongate. Okay, and finally you get a basal cell becomes swollen, and they form the ventral cell, and you have the venter canal cells just above the venter. Okay, 3 to 4 venter canal cells, and the above portion of the archegonium forms the neck region lined by the neck canal cells. Okay, and in the venter you can see the egg, and you have the stalk. The basal cell forms the stalk region of the archegonium. So this is how the archegonium looks like inside the involucre. You have the stalk, you have the, okay, and it is lined by the bracts. These are the bracts. You call it bracts. Even for antheridium, you will have. It is protective in function. So you have the short stalk through which the archegonium is attached inside the involucre or the perianth, and you can see the venter, okay, and the neck portion. It is cover lined by the neck canal cells and the venter canal cells. So you can see 2 to 3 or a cluster of archegonium, 4 to 5 in groups you can see in each involucre. So archegonia are located within the perianth. Only one archegonium is fertilized to produce the sporophytic generation. So normally, like in any other bryophytes, you can see the archegonial, the cover cells, and the neck cells get degenerated and they produce the mucilaginous substances which creates a sticky environment. Okay, and the mucilaginous fluid oozes out and attracts the sperms. So a large number of sperms get collected near the egg neck region. Only one of them passes down to form to fuse with the egg to form the oospore or the zygote or the first cell of the sporophyte. So this is the diploid cell. Zygote is the diploid cell, is the first cell of the sporophyte. So that's what they say. The oospore is the first cell of the sporophyte generation. And the mature sporogonium, once it starts to develop, the mature sporogonium is differentiated into capsule, seta, and foot. Foot, seta, and capsule. You can see it here. It is within the involucre. It comes out from the capsule region comes out from the involucre, emerges out from the involucre. This is the archegonial branch. Okay, this is the archegonial branch. From the archegonial branch, the sporophyte is exposed. The seta foot is inside the involucre. It is not visible. Okay, foot absorbs water and food from the gametophyte. It is the lower portion of the seta, lower expanded portion of the seta. So it is sometimes termed as pseudo foot. And the seta is also quite short. Once the spores mature, they elongate. Okay, they elongate. This is a matured sporophyte. This is an immature sporophyte. Here you have a short seta and you have the capsule which is covered with a jackets layer. Okay, and inside the capsule you can find elaters and the spores. Sorry. This is and the covering of the capsule is called calyptra. Okay, and once the sporogonium matures, the calyptral region breaks and liberates the spores. So how the sporophyte develops or how the sporangium develops, that is how we are going to see here. So the neck of the archegonium shrinks after fertilization and the venter continues to grow and swell. Okay, this is the zygote. So all the other parts of the archegonia are shriveled and they are disintegrated, and this oospore or the zygote, okay, they swell, it swells and secretes a wall. And the first division is transverse division. The lower cell is called hypobasal cell. The upper cell is called epibasal cell. So hypobasal cell does not divide any further. It is going to form the suspensor of the developing embryo. So the function of the hypobasal cell is to form the suspensor. Okay, suspensor of the embryo which absorbs nutrition from the gametophyte. Okay, and the upper cell or the epibasal cell only undergoes both transverse and vertical divisions. It's called periclinal and anticlinal division to produce the jacket layer and the enclosing sporogonial cells or sporogenous tissues, we call it. Okay, so any embryo, the developing sporogonium or the sporogenous tissue layer is divided into outer amphithecium and inner endothecium. So amphithecium forms the jacket, multi-layered jacket cells. Okay, and all the sporogenous cells they differentiate into fertile spore mother cells and sterile elater mother cells. So the sporogenous tissue which is developed in the endothelial part is differentiated into fertile spore mother cells and sterile elater mother cells. So the spore mother cells divides meiotically to produce 4 spores. Each spore mother cell divides meiotically to produce 4 spores, haploid spores. Okay, and the elater mother cells divide to form elaters. Okay, elaters are long and narrow. They are scattered in between the spores. Here you can see this is the developing sporophyte. Okay, and you can see the spores and the elaters, and you can see the amphithecium forming the covering or the jacket for the sporogenous region. Okay, and the amphithecium also produces the calyptra. Okay, this is the calyptra. So the and which breaks at this point only the sporogonium breaks, and point of release of the spores. So this is the perianth. Okay, this is the perianth portion. What is shown here, it is diagrammatically represented in this picture. Okay, the mature capsule is covered by calyptra. Capsule opens by 4 valves along the 4 lines. So you get when the valves separate and the spores and elaters become free, they are released. I can show you a better picture. You can see here when there is a dehiscence of the capsule, it breaks along few lines, valves. Okay, so you get a 4 valved area. One, this is a dehisced capsule, a capsule which has released the spores. So at certain points, the capsule gets broken, the capsule wall gets broken, the calyptra gets broken at certain areas, forming 4 walls. Okay, and this is the calyptra region, the capsule. This is the broken capsule, dehisced capsule. You can see the calyptra, which has released the spores. Okay, the longitudinal section, this is a longitudinal section of the sporogonium. Okay, and when mature, the sporangium opens along the 4 lines of dehiscence to disperse the spores. So you can see the elaters as well as the tetrads, spore tetrads, which are actually the elaters are hygroscopic. They absorb water, they swell up, and they increase the pressure inside the capsule, and thus they weaken up the 4 the 4 valves. They weaken up the 4 lines of dehiscence, as we call it. So the wall breaks at specific points, releasing the spores. So immature elaters do not have thickenings, while mature elaters will have bispiral or trispiral helical thickenings. You can see the thickenings as dark areas in the pictures. So these are some of the diagrammatic representation of the vegetative body. This is the sporophyte, bearing the sporophyte. So this is the archegonial branch. The archegonial branch will have the dehisced capsule with the 4 valves. Okay, perianth or the involucre. This is the antheridial branch. The germination of spore. Spores are haploid. They are the first cell of the gametophytic generation, and they germinate to form a short protonema, leafy protonema. Protonema is the just after the germination of sporophyte, it leads to the formation of a small green body called protonema. It can be single celled or it can be a filamentous structure, and the terminal cell of the protonema divides in all the direction to produce a mass of undifferentiated cells. Okay, one of the cell functions as the apical cell, and through the apical cell only the vegetative plant body is going to be formed. The leafy structure is going to be produced. So it is called a maltose plant body. Okay, and then this protonema only differentiates into the stem-like region and the leaf-like region. So you have a particular you have a distinct alternation of generation which is seen in porella. So it is called heteromorphic alternation of generation, because hetero means different, morphic means morphology, structure. So the gametophytic structure is different from the sporophytic structure. So it is called heteromorphic alternation of generation. So plant body is a gametophyte. It is haploid. It develops antheridia and archegonia. Antheridia produces anthrozoids, sperms. Archegonium produces the egg. Anthrozoids fuse with the egg to produce the diploid oospore. So diploid oospore is otherwise the first cell of sporophyte. Okay, and it has three parts: foot, seta, and capsule. Haploid spores are produced inside the capsule, and spore is the first stage of gametophyte. Excuse the spelling. Okay, the spore is the first stage of gametophyte, is again which is haploid, and they produce the protonemal stage, which is the primary stage of the mature gametophytic thalloid body. I mean leafy plant body. So the protonema gives rise to mature gametophyte, completing the life cycle. So this is the diagrammatic life cycle and the picturized life cycle. This is a graphic life cycle where it is gametophytic generation is represented to be alternating with the sporophytic generation. Gametophytic generation is independent. Sporophytic generation is termed as parasitic because it is totally dependent on gametophyte. So the porella plant is the gametophyte, male plant and female plant, male producing antheridium, female producing archegonium, sperm and egg respectively. After fertilization, oospore, sporogonium, spore mother cells, spores, and protonema. So again, you have a dioecious life cycle, which is seen here. Here you can represent it in diagram form also, where the antheridial branch and the archegonial branch, antheridium, archegonium, sperm and egg, zygote, embryo, and the sporophyte, developed mature sporophyte, where deliberating the spores and spore germinating to give rise to the protonema. So we come to the end of the